today is Dr. Yukari Ishikawa. She's a research associate. Oh, hold on, something just popped in the way again. Sorry. Dr. Ishikawa is a research associate for the Microplastics team in the Environmental Research Group of the School of Public Health at Imperial College in London. She specializes in the development of analytical methods aimed at analyzing environmental contaminants, such as SVOCs, VOCs, and microplastics in air, exhaust gases, waste, and biological samples. Her research career began at the National Institute for Environmental Studies in Japan, analyzing PCBs, PCNs, and PBBs in waste-related samples. This is soon followed by VOCs analysis at the University of Tokyo, POPs analysis at the University of Queensland in Australia, and a personal air pollution exposure study at King's College London in the UK, to her current position of microplastics analysis at Imperial College London. Welcome, Dr. Ishikawa, and would you please begin your presentation?
I'm glad.
No, that's okay. We appreciate you taking time to be with us today. So if you want to go ahead with your presentation, that'd be great.
Thank you very much, Ben, and thank you very much, everyone, for your patience. I'm so sorry about my microphones. I want to share my screen now. This one. Can you see my screen?
Yes, we can see it.
Okay, good.
If you want to start presenting. That's it. Yep.
Okay, great. Okay, thank you very much. I'm Dr. Yukari Ishikawa, a research associate of NIHR, HPRU Environmental Exposure and Health, Imperial College London. Firstly, I would like to talk a brief overview of the analysis of micro nanoplastic environmental samples. Then I will show some examples and challenges of analytical standards for calibration curve and internal standard. First, let me briefly explain about microplastics. Microplastics are fragments of any type of plastic less than 5 millimeters in diameter. They enter natural ecosystem from a variety of sources, for example, industrial waste, car tires, clothes, textile, etc. Microplastics are emerging environmental contaminant across aquatic, atmospheric, and terrestrial compartment. The ubiquitous presence of microplastic raises concern about human exposure and the toxicological potential. Okay. So how is the analysis of microplastic in the environment performed? Since microplastics are present in all types of environments, my apologies today, I have not yet mastered the analytical method for all types of environmental samples. But today, I show an overview of analytical method for air, water, dust, and food here. So first, qualitative and quantitative analysis of microplastics can be broadly divided into two methods, counting the number of microplastics per unit using Raman microscopy or FTIR microscopy, and using pyrolysis-GC to produce weight or volume concentration. Regardless of which method of analysis is in use, the basic principle is to collect the microplastics on a filter. Whenever background matrix interfere with the detection of microplastics, digestion, extraction, or other combination of method is used as required. One of the most important aspects of designing an analytical method for microplastics is filter selection. And it is important to choose the most appropriate filter first, depending on the purpose of your study, the sample type to be analyzed, microplastic particle size you want to capture, and the analytical instrument to be used. For example, in the case of analysis of microplastics in indoor air, glass fiber filter is So glass fiber filter is set inside a glass petri dish placed indoors, where naturally settled airborne microplastic are collected, cut, and analyzed by pyrolysis-GC. So pyrolysis-gas chromatography is useful for identification of involatile compounds. Since microplastics are involatile, their polymer molecules have to be fragmented into small molecules in the pyrolysis unit before being introduced into the GC system. Pyrolysites are separated by GC and detected by mass spectrometry. To optimize the pyrolysis temperature, you can refer to the TGA or DTG data of the polymer you wish to detect. The most popular temperatures for microplastic analysis are 590 and 600 degrees Celsius, although published methods for microplastic analysis use temperature settings between 450 and 750 degrees Celsius. This is the actual polystyrene pyrolysis-GC data measured in our lab. It shows beautiful pyrogram of styrene monomer, diphenylbutane, and the styrene dimer and the styrene trimmer. Our group uses the styrene trimmer peak for PS quantitation. Here is the MS peak of styrene trimmer. Our GCMS includes several MS libraries, so we will use these libraries to identify PS peaks. Also, I use a data book of pyrolysis-GC for manually checking too. This is the book, data book that I love to use. The title of the book is Pyrolysis-GC Data Book of Synthetic Polymers, which is written in 2012 by Tsuge et al. And I really strongly recommend to have this book. This is really amazing book to have. So here are the major qualifying ions used for microplastic analysis and environmental samples. It is important to decide your best qualifying ions by considering the background matrix in your sample. For example, as Okaho stated in his paper, styrene was the most abundant indicator compound from the pyrolysis of PS. However, natural products such as chitin, wood, and fish protein also release styrene as a pyrolysis product. Therefore, styrene trimmer was chosen as the PS-specific indicator compound, which we also use in our lab. Now I'd like to talk about examples and challenges of analytical standard. So as you may know, for GC analysis, a set of standard solution of known concentration is commonly used for making a calibration curve. This is a method that is easy to reproduce, easy to handle, and familiar to everyone. So it can be expected to require less preparation time. However, there is a fatal problem for microplastic analysis. There is no solvent in which PE or PP can be dissolved. This is a very important problem considering that because PE and PP are major plastics in environmental microplastics. Therefore, several alternative methods have been used for making calibration curve. For example, weighing a tiny amount of polymer powder on an ultramicro balance. This method provides you accurate weights. However, it is very time-consuming method. a method using ACE, accelerated solvent extractor, to dissolve polymers in DCM that do not dissolve at ambient temperature and pressure, and obtain a calibration curve by measuring with pyrolysis-GC before the polymer precipitate has been reported. Our group also tried this method, but it was not successful because the polymer did not dissolve well. Method of adjusting the concentration of a polymer by mixing with the other inert solids has also recently been reported. Our group is in the process of creating a calibration curve using this method and testing its reproducibility now. And in addition, a method of mixing polymer powder with the non-soluble solvents. So it's like making suspension and making step dilution series for calibration curve. It's also being tried. From the research papers that have been published, we are gradually learning about the level of microplastic concentration in the environment. As you can see from these data, low concentration standards are required for calibration curve, and challenges are still remain. So now I would like to talk about internal standard for microplastic analysis. As an internal standard, there are two main categories: the use of the mimic chemical or labeled polymers. So if you use a mimic chemical, you decide which chemicals to use and whether the chemical is labeled or not. If you're using a labeled polymer, then choices are whether it is carbon labeled or deuterium labeled, and whether to buy a solution product or solid product. Here I show some example of mimic chemicals have been used for microplastic analysis. In terms of labeled polymers, there are carbon labeled and deuterium labeled polymers, but carbon labeled polymers are very costly. Also, the particle size is unknown. So if you plan to dissolve them in a solvent after you purchase, there is no guarantee actually you can solve the polymer in your solution, in your solvent. So it is because it depends on the particle size. So it is very important you obtain the detailed information of the labeled polymer as much as possible. As deuterium labeled polymers, D5PS and DAPS and others are available now on the market. So here the pyrolysis-GC data of D8PS measured by our instrument. You can see beautiful PS pyrogram here. So the tropylium ions are 91 here, and then 98 are shown. So, but here the A issued. When deuterium-labeled polymer are used, it should be noted that DH exchange may occur in your sample. So here is the data reported by Ryosuke et al. So you can see major MZ peak has been shifted by DH exchange. So they use star and D5 as their internal standard. So so the no matrix here, the major peak, but with the matrix, the major peak slightly shifting. So here the graph showing the total area is shifting depends on the matrix. So after you consider which labeled standard you're going to buy, Also, you have to consider whether you're buying a solution product or solid product. It is important to consider this based on your analytical process, because if solids are purchased, the weighing process should be time-consuming, as I explained earlier. But also, if you if you decide purchasing solution, recovery issue may occur during filtration. Because as I explained earlier, for microplastic analysis, filtration is kind of like a key process for sample prep. So, but this is example of my analysis when I added polystyrene solution as a cleanup spike to my food sample and performed all sample prep and analyzed by Pyrolysis-GC, but I couldn't see any data of polystyrene. So I was scratching my head, and I realized cleanup spike is actually passed through at the filtration process. So now I'm trying to make a precipitation before spiking to my sample. So maybe if you buy your internal standard in solution base, better to consider how you're going to earn your recovery. So here the conclusion. So brief overview of the analysis of micro nanoplastics in environmental samples. Importance of choice of filter or substrate depend on your instrument. And DTG is a useful tool to optimize the pyrolysis temperature. Having a good pyrolysis-GC database is highly recommended. And examples and challenges of analytical standards. The importance of obtaining preliminary data on the approximate concentration in the sample of interest is very important. So the methods for preparing a standard for calibration curve and internal standard remain a challenge. That's all from me today. Thank you very much for your time.
Great. Thank you very much, Dr. Ishikawa. I think we can all see that there's a lot of work that still remains to be able to figure out this situation of microplastics. But I think, there's a lot of researchers that are up and coming and going to be looking at these things. So your initial investigation certainly will be helpful for that. We don't actually have any questions specific to your presentation. So I think with that, we'll go ahead and close. Let me see if I can one last time get this presentation of mine back up and running. And sure enough, it's gone again. All right, so what I was going to say is that brings us to the end of today's Environmental Analysis Isotope Days seminar. I'd like to take another moment to thank all 5 of our presenters for sharing their work with all of us today. I'm sure we can all agree it was a very enlightening day. I would also like to thank everyone who attended today's seminar for your time and attention. To recap, our goals today were to provide you with informative presentations about globally relevant environmental testing programs presented by a group of experts in the field. We hope to demonstrate broader applications for the use of stable isotope-labeled compounds and the scope of CIL-labeled standards. We hope you will all feel more enriched for these presentations. As we conclude this session, you'll be prompted to take a short closing survey. We would greatly appreciate it if you could stay on and take a minute to complete this in order to help us with future seminars. You should also expect to see in the near future a follow-up email with links to today's event and other relevant information. If you have any questions or if you want to discuss CIL products, please feel free to contact us at the email address shown, which it isn't shown at the moment, but it's envsales@isotope.com, or visit our website at www.isotope.com. And that concludes everything from us for today. Thanks again. Have a great rest of your day, and we look forward to seeing you again for Isotope Days 2024. Goodbye.